Hey everyone, welcome to my YouTube channel, and this is Steven. Let's get started with a lot to get through, and given our subject, I don't expect to have to tell people to pay full attention. Uh, in this particular presentation, we will talk about the advanced systematics, specifically in biogeography. This journal is entitled The Genetic Diversity and Geographic Distribution of Genetically Distinct Rabies Viruses in the Philippines. This was written by Saito et al. In the PLOS Neglected Tropical Diseases in 2013. Before we start, let's begin with this news article coming from the Philippine News Agency, and the online article header is stating that love pets, knowledge is the key in your protecting self and dogs from rabies. And the information tells us more about the transmissibility of the viruses to humans, which putting us humans and our pets' life in danger. And according to the health department, almost half of the annual rabies cases in the Philippines involve domesticated pets, not just dogs. This is also an article from 1999 entitled Rabies Cases in the Philippines. This was written by Wallerstein in Manila. It's also a predicted forecast all about the numbers of cases of rabies in the Philippines, which currently ranks fourth worldwide, including the incidence of the disease. And this was projected from 1999 that the problem still exists until the year 2020. And yeah, because of many Filipinos eat dogs, it may also be predicted to helping the spread of the rabies virus. Now, this question: how was a virus with such an extreme life history, with small genome and constrained evolution, become a global threat to human and animal health? Moreover, like many other RNA viruses, the molecular evolution of the rabies virus is inherently constrained. That means the absence of a proofreading mechanism in the virus encoding RNA polymerase, which leading us to high rates of spontaneous mutation. This was from Drake in 1993. Also, while these mutations genetically and might potentially produce in a diverse population of genetic variants upon which a selection might act. It's sometimes called as the quasi-species or a mutant spectrum. Most mutations of your RABV will be deleterious and pleiotropic. Thus, despite constant generation of this novel genetic diversity, there are pathways for adaptive evolution that are likely to be highly constrained in your rabies virus. Historically, we'll take a look at the etymology of the word rabies, which is derived from the Latin word rabies, which means madness, and in turn it may be also related to Sanskrit rabas, which means to rage, and Greeks derived the word lyssa or lod for the root used in the genus name of the rabies virus, which is the Lyssa virus. This is a picture of the miniature of Cantiga depicting two monks hospitalier with rabies being carried before the Saint Mary of Tirina. Historically, this was present, and this is an ancient disease before. Also, a woodcut from the Middle Ages showing a rabid dog. Try to look at these diseases that are present before the occurrence or the discovery of the vaccines for rabies virus. And this is also a map showing the presence and the absence of rabies according to the CDC. And it tells us that the rabies virus is endemic. It means it's regularly found all over the world except for the countries highlighted in green in Australia and Antarctica and some parts of Europe. However, in Latin America, you can see from the previous photo and map, in Asia, the stray unvaccinated dogs are also found. And when I look at the CDC assessment by country, I tried to search the Philippines if what's the current status of the annual assessment of these individual countries for their rabies status to support and inform some of the recommendations as needed by your Centers for Disease Control and Prevention. And it is prevalent that in the Philippines, the rabies surveillance is considered robust. Okay, if it includes some of the methods and the result or constant implementation of different surveillance because of the rampant cases of the rabies viruses in the Philippines, as it was noted from the previous articles. The government has received a lot of publications, example for this one, or submissions satisfying international reporting requirements. And if the data is not available, the most conservative determination for this is applied and surveillance is not considered robust. And there is regionalized efforts that are being done. Some of the matter experts are highly encouraged to contact the CDC if they have to provide additional data considering the updated reported status for their country, for instance in the Philippines. Another photo from CDC with dumb rabies appear depressed, lethargic, and uncoordinated. Rabies, as we all know, is a fatal encephalitic infection which is transmitted to humans from rabid animals, usually with a bite. And various mammals are known to be a natural reservoir. Even exposure to some of the bat species, even without an evidence of a bite, it's also a big risk factor. The virus will enter into your central nervous system by ascending along the peripheral nerves. From the wound, it will be going to its incubation period. It really depends on the distance between the wound and the brain, and the disease manifests initially with non-specific symptoms of body malaise, headaches, and fever. And as the infection advances, the patient shows an extraordinary CNS excitability. The slightest touch is painful, with violent motor responses progressing to convulsions. And contracture of the pharyngeal musculature may create an aversion for swallowing even water. So we call it the hydrophobia. And the periods of mania and stupor will progress to coma and eventually death, typically from a respiratory failure. And this is a schematic diagram showing the sequential steps in the pathogenesis of rabies after an animal bite. Again, you'll see here most likely, since rabies is a neurotropic virus, it spreads along the neural pathways and invades your CNS, where it causes an acute infection. This is also photos coming from the National Center for Emerging and Zoonotic Infectious Diseases in the CDC. Most of us, what we know about the events that takes place during rabies infection has been learned from these experimental models using animals, and these are some of the fixed laboratory strains of rabies virus because they are easier to handle and less expensive, although the events of these models are not closely mimicking the disease under the natural conditions in humans or in rabies vectors. This is also coming from the CDC. You'll see here a number of sequential steps that occur in the after the peripheral inoculation of the rabies from an animal bite. And also this one. Neurons, enlargement of these negri bodies in the brain tissue. And a photo from CDC showing a man with progressive signs of rabies. A young girl about to receive the PEP after being bitten by an animal thought to be rabid. Now we'll direct our attention to the main purpose of this presentation. This is all about the paper, the genetic and geographic distribution of genetically distinct rabies viruses in the Philippines. You'll see in the background Louis Pasteur treating patients for rabies who are manifesting the hydrophobia in the 19th century. So what is the viral classification and biology of rabies? This is what the macro evolutionary dynamics of the rabies virus. This is not found on the paper. I got this one from a paper from Fuchs in 2020, which is the scientific basis of the disease and management. Macroevolutionary, the dynamics of rabies virus is being studied here using the maximum likelihood phylogenetic trees of the nucleoprotein gene, showing the relationships among the major rabies virus lineages. You'll see here the branch colors, which are indicating the suspected reservoir association. The red for Chiroptera, the black for Canidae, the light blue for non-Canidae carnivora, and the green ones is for the primata. And most of it, you'll see here that the Canidae and the Chiroptera or the bats are the major reservoir for the rabies viruses. The New World bats specifically, and your cosmopolitan, your dogs, fox, skunk, and your jackal. This is also coming from Fuchs from 2020. The rates of evolution observed across host-associated viral lineages for the nucleoprotein gene, which are being shown in squares, and your glycoprotein gene, which is shown in circles, in carnivores and bats. This is we will look at in the molecular evolution or the clock rates from Stryker in 2012, Trupin and Pepin in 2017, which contains the details you'll see here of each of the variant and methods used to estimate the clock rates. In this figure, bars represent 95% highest posterior density intervals, and you'll see that the triangles are indicating intervals that were truncated for ease of graphical representation. You'll see that the ferret badger, the skunk north central, the mongoose in Africa, and the ferret badger are the ones which has a lot of substitutions per site per year, and they underwent mutation for these particular genes. And this is the timeline of potential events in the evolutionary history of the rabies virus. These are hypothetical order of key events. Timescales are approximate, and some events could have happened over long time intervals. So this is coming from the 64 million years ago. The Chiropterans evolved. Then the shifting of the rabies lactovirus triggers the evolution of the rabies virus in Oceania, from the hosts going to the old world carnivores. Then there's again continental separation, which isolates the rabies virus in American bats. The ancestors goes extinct from old world bats, and then the dogs were domesticated. Human arrived to the Americas, Europeans colonized the Americas, and now coming from your from old world, the dogs, it transferred into these wild carnivore strains, and there might be a recombination that happened from Chiropterans. It went into some of the indigenous wild carnivore rabies viruses. And this is also coming from the same paper of Fuchs in 2020. It shows here the historic host shifts. Okay, it might not be accurately inferred from some of the contemporary data. In the left panel, you see here two host shift occurred, which is represented by this asterisk. Okay, a virus jumping from species A to species B, and from species B to species C. Okay, and there is subsequent to the second host shift, the virus lineage in species B became extinct. As a consequence, the virus sequence data from the two extant lineages, the A and C, is providing us an incomplete picture by indicating that the virus jumped directly from species A to C. In the right panel, you'll see here. In addition, all of the genetic changes that occurred after the host shift from species A to species B, which is shown in orange, you'll see this line here, is incorrectly attributed to the wrongly inferred host shift from species A to species C. And also for some of the well-studied and putative host shift to diverse orders of mammals. These are the recipient host, the source host, their outcomes, location, timeline, adaptations in some of the key references. Now take a look at the viral classification of the rabies, which is a prototype virus of the lyssavirus genus of the order Mononegavirales of the family Rhabdoviridae. The Mononegavirales is characterized by your non-segmented negative stranded RNA genome, which are encapsulated tightly into a ribonucleate structure, and the Rhabdoviridae are considered or classified as a group based on, you'll see that they have conical bullet-shaped appearance by what we call using the electron microscopy. And as we interfere with some of the legislations accordingly to the previous articles that was posted, there is a paper that has been published in the 2019 about the awareness of the pet owners about this RA 9482, which is the Anti-Rabies Act of 2007. And again, this is not just a this is not just a project by the DOH, but there's a convention of the departments in the Philippines that are acting upon in this Anti-Rabies Act. Okay, so the DOH, the Department of Agrarian Reform, the Department of Agriculture, also with the DepEd, and the LGUs that are acting or solely responsible for the dissemination of this particular act. And in that paper, the results reveal that the pet owners in Magalang, Pampanga in the Philippines had a poor knowledge of the Anti-Rabies Act, and majority were unaware of the existence and of penalties that are imposed to the law to non-compliant pet owners. Also, with this particular structure or the diagram, it shows here a newly discovered virus in Australia that latter killed 2 humans. This was a member of your lyssavirus group, which is related to classical overseas rabies, and this is colored transmission of the electron micrograph of Australian bat lyssavirus. The rabies virus genome structure is being shown here with again, this genome encode only 5 proteins. The virus has the ability to protect itself from what? Ribonuclease digestion, and it must retain its shape ideal for transcription. This is also a schematic representation of the rabies virion. You'll see here that it has a bullet conical shape structure with this particular portion, the glycoprotein spikes, and the R and PM structure. Now, again, the hosts are highly diversified. That includes plants, arthropods, fish, and mammals. And in your rabies and rabies-related virus group, the Lisa viruses is presently composed of the genotype 1, which is the classical rabies virus, and there are 14 other genotypes which are related antigenically and genetically that produces clinical disease indistinguishable from rabies. And the rabies virus genome, again, is approximately 12,000 nucleotides in length and has a molecular mass of 4.6 by 106 kilodaltons. This is also in connection with the National Rabies Prevention and Control Program of the Philippines, implementing a geographically targeted dog vaccination approach to eliminate rabies. And the research from Saito et al. In 2013 was approved and funded by the Research Institute for Tropical Medicine of the DOH in the Philippines. For the materials and methods, we'll take a look at sample collection, FAT, PCR, and nucleotide sequencing. They've used immunofluorescence microscopy that was using fluorescent dyes and can be conjugated with antibodies. You'll see here that the labeled antibodies further are using as probes to detect the locate specific antigen specific to this antibody. And when the immunofluorescence is specifically used to detect an antigen by conjugating your fluorescent dyes with the Fc region of the specific antibody, you'll see here the immunoglobulins against that antigen. So this is what we call as your fluorescent antibody test. And in addition to that, you'll see here, this is not coming from the paper, but I just got here from I got it from a different paper. Immunofluorescence staining of the nervous tissue from rabid animals. You'll see that they are highlighted in this particular figures. For the research paper, the FAT was performed at the regional animal disease diagnostic laboratories. They're coming from every region in the Philippines, and also in the provincial animal disease diagnostic laboratories to diagnose what rabies infection. And when the positive samples were obtained, they were directly shipped to RITM for molecular analysis in accordance with the guidelines for shipping B category infectious substances. As you all know that the cultures of these samples, okay, are intentionally propagated for the purpose of the research, and this definition does not include human or animal patient specimens as defined from this particular protocol. In the FATs, they were repeated to confirm positive results. Brain impression of the smears were sampled in a cold acetone and then air dried. They were flooded with the fluorescent isothiocyanate anti-rabies monoclonal globulins in 1% Evans blue Connor stain, and they were incubated in a humid chamber. They were washed twice with the phosphate buffered saline, distilled in a double distilled water and air dried. Buffered glycerol mounting medium was applied before viewing the rabies antigens under an immunofluorescent microscope. Again, they have also used this RNA mini kit from the Qiagen Incorporated. This is a fast procedure in delivering high quality total RNA in few minutes. Okay, this is also being used for a high performance downstream application of the RNA samples. Consistent RNA yields from small to large amounts will be using this particular kit. Accordingly, this is the primer list of the target genes, we have two genes in particular that are being studied under this paper, the G and the N. Okay, this is the forward reverse primers, and their sequence in 5' to 3' direction, and their positions. In the PCR, the G gene was amplified using the RV7F and RV9RPH, which are generating the 2425 base pairs amplicon. Also, the primers P1 and 304 were used to amplify the rabies virus engine, generating a 1511 base pairs amplicon. They are amplified using the superscript one-step RT-PCR platinum Taq high fidelity polyisomerase. This one. And also Takara is being used in PCR thermal dyes. This apparatus is designed for convenient endpoint detection and analysis of RNA molecules by one-step RT-PCR. Then nucleotide sequencing. They are purified using a big dye exterminator purification kit, and it followed by loading it into the genetic DNA analyzers. The bidirectional sequencing were also performed that uses the primers as listed from the previous table to resolve the complete G gene and the partial region of the N gene. This is your QIA quick PCR purification kit, again used to purify RT-PCR products that are being used prior to nucleotide sequencing. In sequence alignment and genetic analysis, they have been using this maximum likelihood method of your MEGA version 5 algorithm with bootstrap probability calculated for around 500 replicates, and also they are using this general time reversible gamma model in AICC values to model the selection feature of the MEGA5 program. And there are clades and subclades defined when the genogroup comprised more than 4 strains. Again, genogroups consisting of fewer than 3 strains were not considered as clades, and they are indicated by the prefix GR. Okay, all the reference sequences used for the comparative analysis of this particular research were obtained from the GenBank genetic sequence database in NCBI that are shown in the next slides. Sequences described in this study were submitted to GenBank, and the corresponding accession numbers are listed in the links that are available for the complete details of this supplementary paper. For the analyzed samples, so the results of all the 317 brain tissue samples, 51 were excluded because of incomplete epidemiological information, 5 samples, failure of PCR amplification, 21 samples, or sequencing, which are 25 samples. The complete G gene has 1,572 nucleotides, and partial G gene were determined for 233 and 30 strains respectively. Showing here a phylogenetic tree partial G genes from 266 strains was obtained in the supplementary figures, and because of the low bootstrap values, only samples from complete G genes were included in the analysis. This is the first figure of the phylogenetic tree of complete and partial G genes from the 235 Philippine rabies virus strains. Again, you'll see here colors that are indicating the different clades of your green, blue, and orange. This stands for the major islands in the Philippines, the Luzon, the Visayas, and the Mindanao. And dots on the map indicates the number and collection sites for each of the samples. One dot is equivalent to one sample. Bootstrap values of greater than 90% is presented in this particular tree. On the other hand, the figure 2 is comparing the phylogenetic trees of rabies viruses in N and G genes. In your N, partial N gene, there are 1,124 nucleotides, and your complete G gene has 1,572 nucleotides. And these are using the 57 virus strains from the Philippines and other Asian countries. These trees are being constructed with the maximum likelihood method, and bootstrap values were calculated from 500 replicates. These trees are showing you the three major clades in color: Luzon, Visayas, and Mindanao, and the two distinct strains coming from the Mindoro, this one, the GRMD, and the GRSL from South Luzon. And you'll see here that the red circle shows the Philippine strain from the cluster that was previously named Asian 2B by Gong et al. This is also a figure from the paper, The Geographic Distribution of the Clade Luzon. This one is a phylogenetic tree from the G gene that are from 235 Philippine strains, was again constructed using your maximum likelihood method, and bootstrap values were calculated from 500 replicates. The condensed tree in this diagram with the cutoff value of 80% is presented, and the Clade L is enlarged. There were 9 subclades L1 to L9 and 3 genogroups, the GRL1, GRL2, and GRL3 in the Clade L. Let's move with the subclade L8. This is a G gene again from the 235 Philippine strains that are constructed using the maximum likelihood method, and this is a condensed tree, okay, of the subclade L8. Geographic distribution of the subclade L9, and you'll see here the existence of the genogroups L9B and LNA. For the Visayas and Mindanao, this again coming from 235 Philippine strains being enlarged. Clade number 5 was subdivided, or Clade Visayas is subdivided into two subclades, the V1 and V2, and the Clade Mindanao comprised of only one subclade, M1. Bootstrap values of more than 80% was observed. And to end this presentation, rabies continues again to be a major public and health problems that is in the world, but also in the Philippines, where 200 to 300 human cases are reported between 2001 to 2011. And understanding the phylogeography of the rabies viruses is more important for establishing a more effective and feasible control strategy. We have studied the detection of the 3 major clades and 2 distinct genogroups of rabies viruses in the Philippines. And our data suggests that viruses for each of the clade and subclade evolved independently in each area without frequent introduction into other areas. And an important implication of these data is that geographically targeted dog vaccination using the island group approach would be an effective method to control the rabies in the Philippines. Every 10 minutes, someone dies completely preventable death from the rabies virus. With access to quality and effective medicines and vaccinations, we researchers can stop this. Thank you.